Hello and welcome to my digital poster presentation for the ISMRM 2021, where I'll be presenting my research abstract, Cardiac Metabolism Assessed by MR Spectroscopy to Classify the Diabetic and Obese Heart, a Random Forest and Bayesian network study. Knowing the increased risk of heart failure associated with obesity and type 2 diabetes, we are motivated to understand the cardiovascular remodeling and metabolic changes caused by these diseases. One such metabolic change is a decreased PCR to ATP ratio, which can be non-invasively assessed using phosphorus 31P MR spectroscopy. A second is an increased myocardial lipid accumulation, which can also be quantified using proton MRS. Using the data collected in our institute, we wanted to answer the question of whether or not these metabolic parameters can significantly differentiate between an obese or diabetic heart versus a healthy heart. And if so, how does it compare to global function parameters? To do this, we utilize machine learning to investigate this in a more explanatory way. Thus are the two experimental goals of this project, which we'll delve into shortly. After data cleaning, our data sets featured 197 data points, 74 healthy, 55 obese non-diabetic, and 68 diabetic. We performed 3 classification experiments: healthy versus obese, healthy versus diabetes, obese non-diabetic versus obese diabetic. This was to investigate cardiac impacts of diabetes independent of obesity. For each, we used a 25-75 test train split, training a random forest classifier, taking out definitional variables where relevant. We use entropy and purity as a performance metric with tenfold cross-validation. Among various feature importance metrics, we chose SHAP as a state-of-the-art, whereby to determine the importance of a single feature, the outcome of each possible combination of features is considered. A model is trained for each and the impacts are averaged out, resulting in a more accurate and independent measure of a feature's predictive importance. Finally, we wanted to not just interpret which parameters were most informative, we also wanted to understand why, and particularly how these parameters interrelated to one another. For this, we generated Bayesian networks for each experiment. Briefly, a Bayesian network is a probabilistic graphical model where each node represents a random variable and the edges represent conditional probability dependence, implying a causal influence. Famously, correlation is not causation, and you may be aware of Simpson's paradox as a prime example. Bayesian networks allow you to the detection of this and many other benefits, so it's not surprising that they have been applied to medical diagnostics since the early 90s. However, limitations in computational approaches have hindered its application, but luckily, recent developments have found mathematical solutions to this intractability, as outlined in this recent breakthrough paper, the Noether's algorithm, which we have applied in our project. Looking at the results, we found that the highest accuracy model came from the healthy versus diabetes classification, with a holdout accuracy of 91%, suggesting that the cardiac parameters deteriorate most in diabetic patients compared to obesity, which is consistent with the statistics previously shown. To distinguish between healthy and diabetes, besides age and BMI, we see that having a high visceral fat is the next most important feature, then a low stroke volume, high cardiac lipids, and a low PCR ATP. For healthy versus obese, these metabolic parameters are ranked highest, followed by systolic blood pressure and cholesterol as expected. Controlling for obesity, visceral fat, and metabolic parameters are once again the most impactful. These results suggest that varying degrees of cardiac function can be observed in obesity and diabetes, while low PCR ATP ratio and high cardiac lipids are almost always present. From the generated Bayesian networks, we see the algorithm has learned quite a representative set of relationships. In a nutshell, here are a few key observations. There is an implied causal relationship between an increased visceral fat mass and LV mass and concentric LV remodeling. This is consistent with theories of visceral fat linked to subacute inflammation in diabetics. Next, increased LV mass causing decreased PCR ATP, further supporting the results from the SHAP and theories of hypertrophy. Finally, obesity causing increased myocardial lipid concentration. In conclusion, our results reveal a high predictive impact of metabolic parameters with theorized causal links to energetic impairment in diabetes and obesity, thus alluding to its potential as an early diagnostic tool for the onset of cardiac disease. Thanks for listening, and many thanks to our collaborators and sponsors.